Good afternoon, everyone. Thank you all for joining us this afternoon. So we're going to be talking to you today about 25 years of long-term monitoring on fringing reefs around inshore islands in the Great Barrier Reef Marine Park. I'd like to start by acknowledging the Bindal and Woolgooroo Kabar people as the traditional custodians on the land where we are today, and pay our respects to their elders, past, present, and future. We also acknowledge the traditional owners of the sea country where we work, and we have a full list of these groups at the end of our talk. This webinar is being recorded and will be available on the Tropwater website after this talk. And I can't advance. So joining me for the talk today are my colleagues, Gemma Galbraith and Eva McClure, who've been working with me on this project for the last 4 years. I'm first going to give you a brief introduction, then Gemma will share our latest results and talk about long-term trends in coral trout abundance, biomass and size structure, and the effectiveness of marine reserves. Coral trout species are among the most highly targeted fish species on inshore island reefs. There's another group known as the secondary target species, which Eva will then talk about, with some examples of some of the analyses that she's been doing on the effects of marine reserves, looking at the size and age of different reserves, the influence of habitat and environmental variables, and changes in marine park zoning and recreational fishing regulations. Next, I'll run through long-term trends in coral cover and give some examples of the effects of coral bleaching, flooding, and cyclones. Then I'll wrap up and I'll talk about the need for continued monitoring of these inshore island reefs. We'll have a Q&A at the end. If you want to ask questions, you can ask them at any time in the chat, or if you raise your hand at the end in the Q&A session, one of our comms team will then unmute you and you can ask your question. At the webinar, you'll be directed to a quick survey, and we'd really appreciate your feedback. No-take marine reserves have been embraced around the world as a management tool to protect exploited species and for biodiversity conservation. And we have lots of evidence from all over the world for the effectiveness of marine reserves, with increased numbers and sizes of target fish species within reserves compared to outside reserves, and as well as spillover of adults and supply of juveniles from reserves to fished areas. They can also act as biodiversity conservation areas, protecting species and habitats, and can enhance resilience, which is the ability to resist or recover from a disturbance like coral bleaching or a crown of thorns sea star outbreak. But increasingly, we're seeing that marine reserves alone are not enough to protect coral reefs from widespread severe disturbances that are predicted to occur more frequently as our climate changes. Inshore reefs in the Great Barrier Reef Marine Park are already under increased stress due to terrestrial runoff and sedimentation from human activities on land. And this makes them more susceptible to other stresses and disturbances and can hinder their recovery. On the Great Barrier Reef, one of the key management tools is our amazing network of no-take marine reserves. Since the rezoning of the marine park in 2004, we now have 33% of the coral reef area of the park in no-take zones. The Inshore Island Reef Monitoring Program was established in 1999 with the aims of examining the effects of marine park zoning and tracking changes in reef fish and benthic communities over time, and evaluating the effects of impacts like coral bleaching, cyclones, and flooding. The key players in the first 20 years were David Williamson, Richard Evans, and Danny Ceccarelli, and Eva, Gemma, and I took over in 2022. It's hard to find money for monitoring, and Dave and colleagues did really well to keep this going for as long as they did on 2 to 3 year grants. However, they weren't able to find funding to keep this program going beyond 2019. So with Dave and Danny's help, we've managed to get some funding from the Reef Trust Partnership for the last 3 years to restart the monitoring. And we've expanded the spatial scope of the monitoring to include 4 new locations. So we now have 4 long-term locations. These include the Whitsunday Islands, which have been surveyed since 1999, the Palm Islands since 2000, the Keppel Islands since 2002, and Magnetic Island since 2004. We've added 4 new locations since 2022, the Turtle Group in the far north, the Franklin Islands just south of Cairns, the Family Islands off Mission Beach, and the Cumberland Islands off Mackay. We now have 145 long-term monitoring sites around 38 islands. These sites include areas of reef in fished areas and in no-take reserves or green zones. We do underwater visual surveys of fish and benthic communities using transects. We survey 5 transects at each site, 50-meter transects. So we're covering an area of a length of 250 meters along the reef. The map on the left shows all of our monitoring sites in each region. All of those white dots is a monitoring site. We have sites in the yellow, dark blue, and green zones. And on the right is the guide to what's allowed in each zone, which many of you will be familiar with. There used to be unregulated commercial fishing on these inshore island reefs before the Great Barrier Reef Marine Park was established, but then the commercial fishing moved further offshore, and now there's only recreational fishing on these reefs. But the fishing pressure around these islands from recreational fishing is quite high. These islands are very important for tourism and recreational fishing. For example, over 500,000 tourists a year visit the Whitsunday Islands, which is about half the total number of tourists visiting the Great Barrier Reef. And this monitoring provides important information on the health of these reefs and the fish communities. So these sorts of surveys, full fish and benthic community surveys, are the most detailed in situ survey methodology. This program is the only long-term monitoring program on the Great Barrier Reef focusing on inshore island fringing reefs and surveying both fish and benthic communities. So this program complements the AIMS long-term monitoring program, and we feed our data into their database. AIMS do have a few survey sites around these islands, but their focus, the long-term monitoring program's focus, is on covering as much of the Great Barrier Reef as possible, and they have lots of sites further offshore. This project focuses on inshore island reefs, and we survey much more intensively around these islands. And the fine-scale detail of this methodology allows us to extract numerous essential ecological metrics of reef health and biodiversity for conservation and management. For example, fish species richness, the number of species, fish density and biomass, coral cover, benthic complexity, etc. Here are just a couple of papers that have been published based on data from the first 20 years of surveys, and you can have a look at those if you're interested in them. And now I'm going to hand you over to Gemma Galbraith to continue the talk.
Excellent, thanks, Maya. Yeah, so there is an awful lot you can get out of monitoring data that's collected by this project, but I'm going to start off with just showing you some snapshot sort of exploratory analysis and results from some of those key metrics that Maya's just mentioned. So here we have got box plots. These are from the most recent 2 years of the Inshore monitoring project. On the left there, you've got total live hard coral cover. And then on the right, this is fish species richness at the site level. And yeah, this is really the sort of thing that we often start out a lot of our analysis with. So to get a really nice overview of some of these regional differences and also differences between green and fished zones. And I think without getting into too many specifics of these figures, what you can really see here is that there is quite a lot of variation between these regions. So both the long-term 4 regions and the new regions, new to the 2 most recent years of this project. So, for example, in the Franklin Islands, in the top left of each panel, you can see there's actually some really remarkably high coral cover in both green and fish zones across both the project years. And then to the Cumberland Islands, less coral cover, but interestingly, if you then look at those same regions for the fish richness, they're really quite similar. So yeah, this is a really sort of key starting point for us starting to think about, what's driving these regional differences, and sort of understanding how that variability, should be considered for a lot of the information we can then provide in terms of regional management. Again, another set of plots with some of those really fundamental metrics we use. So here we have, this is now the biomass of large predatory fish, and then specifically within that total biomass of coral trout. And this is a really just neat example of where you can see a very clear marine reserve effect. So consistently, I think in most regions here, we have a statistically significant higher biomass of both predators and coral trout in green zones compared to fish zones. I think the Palm Islands is probably the only region where we've got a little bit of similarity between the zones, which is interesting in itself. But yeah, again, that regional variability and sort of recognizing that and understanding that across the spatial scale that we've been able to cover in this expanded phase of the project is, I think, is really interesting and important. So if we just move on from some of those univariate metrics and sort of think about what else this monitoring data can let us look at and analyze. This is a multivariate analysis, a distance-based redundancy analysis that is essentially looking at fish community structure. So because we do count and identify every fish within our transects, we can really start to build up a much sort of richer picture of the communities that that populate these inshore reefs. So in the figure with all the colored dots to the left there, each of those dots is a transect. The analysis separates these out in terms of their dissimilarity. It's a little bit counterintuitive, but basically transects with a similar community structure will cluster together. And that's exactly what we see in our regions. So some really quite cool and distinct clustering of the regions. And then if you can see that we've got both open and closed dots, those represent the fished and green zones within each region. And again, this is just some sort of preliminary stuff, but you can really sort of see in some of those regions how the fished and the green zones do also separate out within a region. And then because the methodology also collects benthic data, so we have paired fish and benthic data per transect, we can also bring in some habitat variables to sort of start looking at, what is driving this separation, what regions, are driven along those arrows you can see there leading to things like algae or maybe more coral, different types of coral. So yeah, another really cool example of just the breadth of the method and the data that we're able to collect. So that was obviously a very quick snapshot of some of those results from the last 2 years of the project. I think we can see how valuable those new regions have been to add to the monitoring, really increasing the spatial coverage of the project and the data. But as Maya said, these last 2 to 3 years of funding have allowed us to extend the time series from not just 2 years, we're getting now 2 decades of data, not just from 4 regions, but from 8. And yeah, this is really letting us start to ask some pretty significant questions at that larger spatial and ecological scale. And one of those that we are particularly interested in is the effect of zoning on coral trout, so marine reserve effects for coral trout. And this analysis does use our sort of 4 focal long-term regions, so Palm, Whitsunday, Keppel, and Magnetic Island. And this sort of time series plot you can see here, just on the right, this is just sort of 3 time series that we've been considering, sort of based on the rezoning in 2004. So the 4 years preceding that, we then have the following decade, 2005 to 14, and then the most recent decade, 2015 to present. And just the sort of I guess relative abundance of 2 of the 2 main trout species on these inshore reefs. So that's Plectropomus leopardus and Plectropomus maculatus. This is just a summary of density across each of those regions, and you can see that each region's kind of got a little specific ratio of these species going on. So the Palm Islands there has quite an even mix of P. mac and P. leo. The Whitsundays tend to be more dominated by Plectropomus leopardus, and conversely, the Keppels there dominated by Plectropomus maculatus. So yeah, just getting an overview of the coral trout populations by region there. And yeah, again, because we do have this incredible time series, it lets us start digging into things a bit like this. So some of you may have seen these figures before at various other presentations, but this is the latest data. So from 99 in the Whitsundays through to all regions in 2024. And those dashed lines there are the 3 time periods that I just mentioned. And what we've been doing is looking at the reserve effect, so the comparison between density, or biomass in this case here, biomass of coral trout between fished and green zones as a ratio. And that's what those effect size plots are, those half eyes on the right there. So you've got biomass in green and fish zones as the lines in green and blue. And you can see, again, a lot of variation throughout time and across our regions. But a really key takeaway and a really cool part of the analysis that we've been able to do is look at how this effect size, this reserve effect, has changed over time. So here in the Keppel Islands, you can see that that effect is really quite consistent. You've sort of got about twice as many trout, twice as much trout density in on biomass in green zones compared to fish zones. In the Whitsunday Islands, this is a really cool increase in the effect size. So from pre-zoning over the 2 subsequent decades, we're getting an increased reserve effect. So higher biomass of coral trout in green compared to fish zones. And then again, just back to that variability among our regions. So at the Palm Islands, we're actually seeing a decrease in that reserve effect. And this type of analysis just always tends to generate more questions than answers. So that's definitely something that we're sort of trying to look into with a lot of those other habitat variables that Maya mentioned this project collects. So yes, our counts let us estimate size of all fish in our surveys. And as we've just seen from those biomass plots, how that translates to biomass, which is particularly important, obviously, for things like fisheries management. But with those size estimates, this data set also gives us a really unique opportunity to explore sort of the changing size frequency of fishes between island groups and over time. And I'm going to pass you over to Eva now, who's going to present a little bit of this analysis that we've been working on with the coral trout.
Thanks, Gemma. Hi, everyone. All right, so I'm going to talk to, as Gemma said, some analysis we've been doing in trends in size structure over time, over the lifetime of this data set. So we looked at trends in size structure for each species separately. One of the reasons being, as Gemma pointed out, they occur in different abundances in the different regions. So on the left-hand side, you can see we've got Plectropomus maculatus in our 4 regions, Keppels, Whitsundays, Magnetic and Palm, and then P. leopardus on the right-hand side for the Whitsundays and the Palms. And what I want to point out here, well, first of all, this kind of plot is called a joy plot for, I think, obvious reasons, because they're lovely, but they allow us to do some cool things. So first of all, we can see that the green sort of density plots are representing our green zones, and the blue is representing the fished areas. Across the x-axis, we have the total length, with the dotted line representing the minimum legal size of trout, which is 38 centimeters for both species. And with these joy plots, we can also do things like follow cohorts through time in some regions. And I don't know if my pointer works for you guys, I hope so, but in the Keppels, for example, from 2004 sort of to 2008, you can kind of see this wave go through the fish in the blue zones with increasing size. Okay, so we can do this in a more statistical sense as well. This is a much more sort of traditional way to look at trends in size structure. So we're just going to focus on Plectropomus maculatus at the Keppels here. So we've got our 3 time periods that were mentioned earlier. We've got pre-rezoning in the top panel, sort of just looking at this left-hand side top panel, then the first decade after rezoning and the second decade after rezoning. On the right-hand side, we've got the modelled cumulative frequency. Across the bottom, on both plots, on the x-axis, we have the total length, and again, the dashed line represents the minimum legal size of a trout. And I've sort of split it out. We've got panel D here shows modelled cumulative frequency of both blue and green together, so we can kind of compare them. The different strokes for the lines represent our different time periods. And then we've got the panels below being green and fished areas. So what you can get out of this is you will see the numbers here next to on panel E. And what we've seen is that in our first time period, so before rezoning, the proportion of fish that were below the minimum legal size was 0.67. And since then, we've had in the decades since then, we've had decreases in the fish that are lower than minimum legal size to 0.55 and 0.33. So this basically means that as time goes on, we have more larger fish in green zones. And we might have expected that in green zones, but one of the really interesting things was that this happened in the fished areas as well. So before rezoning, we saw that the proportion of fish below minimum legal size was, 80% of fish were below minimum legal size, but through time that's reduced to kind of 65% and 55%. So this is a really nice kind of signal through time. And, why might that be happening in blue zones? There's potentially a few reasons for this. It could be that there's less fishing pressure as time goes on, but we're rather certain that's not the case. And so this might be a nice indication of spillover, for example, from green zones into fished areas. So another thing that I wanted to look at was we've been talking about trout, and they're probably the most famous of the line fishery targets, and there's specific catch quotas that manage trout, but a lot of the other highly desirable targets of the reef line fishery, often targeted by wreck fishers, are these sort of secondary targets or other species, and they include your things like your emperors, a lot of your snapper or lejanid species, and yeah, while fishing pressure is assumed to be quite high on inshore reefs, the sort of relative roles of reserves and habitat effects and environmental effects hasn't really been looked into. And so one of the beautiful things about a data set like this over the long term regions across the GBR, we can sort of start asking these questions of, what is fishery independent data for species that are targeted by fishers. And so I'm just going to take you through some of the rationale of an analysis that I've been working on. We're going to focus on Lejanus carpentatus, and I apologise to anyone here who might be a wreck fisher who thinks, boring, no one targets these guys. I may be about to prove you wrong, but also they just provided a really nice model species for us, because we've, obviously, well, I haven't surveyed for the 25 years, but with 25 years of data and lots of counts of this species in particular, it provides us with a really nice model. So what we do is we take the biomass of this species. We know that environmental and habitat variables can also be really important in influencing the biomass of not just this species, but reef fish generally, as has been discussed. We incorporate that into machine learning techniques, gradient boosted machines, boosted regression trees in this particular instance, to identify the most influential environmental and habitat variables. Then we can incorporate those into another model as covariates to really ask questions about our reserve effects. So we can account for the variability that these other the environmental and habitat variables produce, and then ask questions about marine reserves. And so we can do this over both the long term and the short term, looking at just median biomass. Are there more fish in green zones compared to fished areas? And does the size and the age of marine reserves, for example, make a difference to biomass once we've accounted for habitat variables and environmental variables? And so here we have our beautiful Lejanus carpentatus, and we see that just when considering the environment and the benthos, the things that explain the variability in biomass include the general fish community diversity and the benthic diversity as being two of the big ones, and then things like the exposure of the reef. So we've got this routine wave exposure, which gives us an indication of how exposed the site is to the prevailing southeasterly trade winds, for example, and the habitat complexities. This is a combination of coral cover, the slope of the reef, and the general complexity. So we can incorporate these variables as covariates into a model asking questions about reserve effects. And what we see is that across the lifespan of the data set, so across 25 years, averaging across all sites, we see that there are more Lejanus carpentatus in areas closed to fishing than there are in areas open to fishing. That's in this top panel here, and on the right hand side we get this sort of half-eye effects plot that shows us that actually there's somewhere between 1.4 and 1.5 times the number of, or the biomass of Lejanus carpentatus in green zones compared to fished areas. Then we can also ask whether larger reserves and older reserves have more biomass of these fish, and generally we see that they do. So this, the panel on the bottom left-hand side is green zone size in hectares and the responsive biomass of carpentatus, and we see that it increases with increasing reserve size to a point, and that's probably because nearly all of our reserves are below this kind of 7,000 hectare size. And then when we look at the age of reserves, we see a really steep increase with increasing age with biomass. So let's move on to some more exciting, potentially, fish species. I will just wrap up Lejanus carpentatus for the more recent years where we expanded beyond just those sort of 4 time points. And we can really get into the regional kind of effects. And we can do this to sort of ask questions around, are some of these species potentially more heavily targeted or more likely to be retained in some regions more than others? And so the different colors here in these effects plots represent the amount of evidence that we have that is the case. And the blue and the black is really what you want to look at. And we see that we have either quite strong evidence or quite good evidence that for Lejanus carpentatus, that there is a reserve effect in most of our regions, the Keppels, the Cumberlands, it's a new area, Magnetic Island and the Palms. And then for one of these other species, the black spot tuskfish, for example, we see that there's potentially some regional differences in either where these guys are targeted or where they're retained. And so the Keppels, the Cumberlands and the Whitsundays seem to be the areas that people may have a preference for going after these fish. We can do other things as well. Look at these larger emperors, for example, and look at the kind of interaction between structural complexity and age. And what we're seeing is that, you know, old reserves are great, but largely there needs to be habitat for the species as well. And it sort of really just highlights that both of those things are quite important. And the effects plot shows that, perhaps unsurprisingly, these fish are, you know, desired everywhere, as there's 1.5 to 1.7 times the biomass of these fish in green zones than in fished areas. I've also been looking at red emperor. You might ask why I look at red emperor on an inshore reef. For those who know about red emperor, you know, they tend to spend their juvenile years or their subadult years around the inshore reefs before moving offshore, where they might be captured in fisheries in between the reefs. But just a cool thing that we've been looking at is showing that, you know, this is back to the joy plot, sorry, and we see that we had an increase in the minimum legal size from 45 centimetres to 55 centimetres. And in the Keppels, this really made a difference. And now we're seeing larger fish in green zones there. Generally, the fish seem to be smaller in the other regions, but we're seeing, if we just have a look at this effects plot, a reserve effect in around the inshore reefs in the Keppels, where they're getting to minimum legal size now and can be taken by fishers. All right, I'm going to hand you back now to Maya. Thank you.
Thank you, Eva. So I'm now going to be talking about long-term trends in coral cover. So these are some figures showing long-term trends in coral cover over time for our 4 long-term regions. They're similar to the ones that Gemma showed you earlier for coral trout, with the blue line for fished areas, green line for green zones, but this is for coral cover. So coral cover has varied through time in both green zones and fished areas at all of these locations, and there are no reserve effects on coral cover. So the thing that has been affecting coral cover, so coral cover has varied both in green zones and in fished areas. There's no reserve effect. And there have been a series of disturbance events that have influenced, that have affected these areas. And I've got them there in these little symbols for coral bleaching, cyclones, and floods. Each region has its own history of disturbance involving these cyclones, flood plumes, and coral bleaching events. And some of these disturbances, not all, some of these disturbances have resulted in declines in coral cover. And there have been periods of recovery following these disturbances. The 2 major events that have caused declines at the Palm Islands are bleaching in 2005-2006 and Cyclone Yasi in 2011. The Keppel Islands are similar, with bleaching in 2005-2006 and not Cyclone Yasi, but flood events in 2011 and 2013. And for the Whitsundays, it was Cyclone Debbie in 2017. So coral cover remained relatively constant over time, and then it just had a massive decline after Cyclone Debbie. And because Cyclone Debbie was by far the biggest impact on any of these island groups, I'm just going to talk to you a little bit about that in the next few slides. So Cyclone Debbie was a slow-moving Category 4 cyclone, which hit the Whitsundays in 2017 and absolutely devastated large areas, both on land and underwater. Now, cyclones are relatively common on the Great Barrier Reef, and the expectation is that recovery is quite rapid. They free up space for new corals to colonize on reefs. But it hasn't been the case in the Whitsundays, unfortunately. So this is just a couple of pictures to show you the aftermath of Cyclone Debbie. The pictures on the left are from the AIMS marine monitoring program, showing lots of live coral before the cyclone, same area after, completely reduced to rubble and the sort of thick, sludgy sediment. The picture on the right is a photo from a survey just after Cyclone Debbie in our program, showing a massive Porites coral just snapped off. Its base was about 1 meter across, and it snapped off and it's upside down at the base of the reef. So this graph shows that trend in coral cover since 1999, and you can see this massive decline after Cyclone Debbie. We've got a paper in press on this, and we just looked at the 3 years before Debbie, the 2 years just after Debbie and the year after. We're calling those the impact years, and then the 2 years after Debbie, so 2022 and 2023. And I've split the corals into massive and encrusting corals and the complex corals, which are the corals that you'd expect to be more affected by such a destructive cyclone. So both massive and encrusting corals and complex corals declined immediately after Cyclone Debbie, but that decline was much greater for the complex corals. And there was no significant difference between the impact years and the post-Debbie years. So there hasn't been recovery, although you can see there's sort of the beginnings of recovery. There hasn't really been much recovery since then. We examined the effects of this coral decline on 2 fish families, the damselfish and the butterfly fish. And these are the 2 families that are likely to be affected the most by declines in coral cover. And we found that for both of these groups, species richness and density declined after Debbie. For the damselfish, there was an increase in density back to almost pre-cyclone levels. Both density and species richness recovered, but for the butterfly fish, there was a further decline. And we did some analyses looking at the community structure, and we found that, I don't have time to show you that today, but we found that there was a shift in community composition for both of these families from more coral species higher proportion of species that live in live coral to a high proportion of species that live in algal areas or rubble. We looked at species level trends for a few of the most abundant species, the butterfly fish on the right and the butterfly fish on the left and the damselfish on the right. And all of the species had significant declines following Cyclone Debbie. And I just wanted to point out these 3 species. So these 3 species live either on algal areas on reefs or rubbly areas. So you wouldn't expect a decline in coral cover to have an impact on these species. And we think that the decline in these species after Debbie is direct mortality. And there haven't been any studies prior to this that have shown that there has been a direct effect of a cyclone on fish. It's always an indirect effect through its effect on the habitat of the fish. The other species are all species that live in and around live coral, and their numbers have continued to decline or have stayed relatively low over time. So from one sad story to another, sorry. Corals, talking about now the Keppel Islands, and so corals bleach when they're stressed, and it's not just higher temperatures that cause bleaching. Reduced salinity can also cause bleaching and cause mortality. So the biggest impact on corals in the Keppel Islands has been 2 flooding events in the Fitzroy River, one in 2011, one in 2013. And this resulted in the greatest decline in coral cover that we've seen in this region. And some of the sites still haven't recovered. So that picture on the top right there is a macroalgal covered reef that was impacted by this flooding event. Some reefs fared much better. The picture at the lower right shows a relatively healthy reef with very high coral cover. But unfortunately, when I took this photo, we were at the beginning of the mass coral bleaching event on the Barrier Reef in February last year. So you can see the corals there are sort of starting to go a bit pale, a bit pink. And I've put this line in here to show you that that's when the bleaching event happened, and we don't have recent data to show what the effects of that bleaching was, because the funding for this project ended at the end of last year, so we haven't been able to do a follow-up survey at the Keppels following the bleaching last year. We were also at the turtle group when the bleaching event started, just after our trip to the Keppels. And in that drone photo on the top right, you can see the little white spots. Those are individual plate corals that are completely white, completely bleached. Down deeper, it wasn't as bad. And so the photo on the lower right shows you a deeper area of reef that's still bleached, but not quite as badly affected as the shallow areas. And again, we haven't been able to do any follow-up surveys at the turtle group. Finally, I'll talk about the Palm Islands. So the greatest impact that the Palm Islands Palm Islands reefs have had over our monitoring period has been Cyclone Yasi in 2011. Coral cover has increased steadily since then, and it's been the highest it's ever been in the last 3 years. Major flooding in March this year caused freshwater stress and sedimentation on these reefs, particularly in the shallow areas of the reef. So we were able to get some funding to go back to the Palms this year in August and do some follow-up surveys. And we found that although coral cover had declined, at least at the depths that we survey, which is the midpoint of the reef, it wasn't as severe a decline as we had feared. I thought it was going to be a very depressing trip, but it wasn't too bad. The shallow areas were pretty dead. You can see in these photos in Pioneer Bay, a lot of dead corals on the reef flat, and these giant clams also all dead. But deeper on the reef, it was a nicer picture. So I've put these photos in just to show you some examples. So top left is a large Porites colony in shallow water. Most of it's dead. There's a few live bits at the bottom that will probably grow back if it survives. And then a little bit deeper, you have a mixture of partially dead and fully live colonies. And then the acroporas, so these are the branching corals, mostly dead in the shallows. This is a bit deeper. There's a large rubbly patch that had a few live fragments remaining. And in contrast, that picture on the lower right is from one of our exposed sites on Pelorus Island. So the sites on the exposed sides of the island fared a lot better. And this is a photo from the reef slope on one of those sites showing a nice healthy reef. So it was really lovely to go and get in the water and see that there's still some amazing reefs at the Palms. So to conclude, we found that coral trout density, biomass are consistently higher in green zones compared to fished reefs. And we've seen a difference in size structure between green zones and fished reefs, and also across time periods. The secondary target species work, we found that there's an effective green zone that varies with species and with region. It may reflect regional fishing pressure on particular species. Analyses are ongoing to tease apart the habitat, environmental, and zoning influences. So watch this space. We'll have some papers coming out in the next few months, hopefully. Finally, we've seen no evidence that green zones prevent or reduce impacts on coral reef habitats. Each location has its unique history of disturbance and recovery due to events like coral bleaching and flooding and cyclones. And none of this information would have been available without long-term monitoring. And our data shows the importance of studying different regions. So there really is a need to keep this project going. And if anyone out there has any ideas or wants to give us some money, please get in touch. I just put this in there to show you how our data has informed other research. So this is from a modeling study that used our data plus a whole lot of other data to show that 1 in 2 coral trout that are caught in blue zones are from parents in green zones. And so this is a real win-win for both conservation and management and fishing. These are just a couple of examples of posters about the benefits of green zones that are based on results from this program and a program on coral trout population connectivity, just to emphasize that long-term monitoring is important, not just for informing management, but also for contributing to community awareness initiatives. And thank you all for your attention.